Thank you everyone for showing up after lunch. Now we turn to a different topic, which is weighted gene co-expression network analysis. And just to give you a high-level view of where we are, in terms of our terminology, WGCNA is really an unsupervised machine learning analysis. And it was originally developed for analyzing really genes based on expression data. And that's why you notice gene co-expression. So it looked at, for example, to cluster genes and to study their network properties. But the underlying mathematics certainly allow you to use the method in very different contexts. So not just for clustering genes, we applied it to many other domains. And so, well, without much ado, let me mention that apart from the web page that will be posted as part of this course, there are other web pages. For example, we teach a workshop, and also there is a free book where you can learn more. But in any event, so today I will teach you how we construct a weighted gene co-expression network, why we use something called a soft thresholding approach, how to detect network modules, and so on. So remember what a standard differential expression analysis does. Imagine, so you study 50,000 genes, they may correspond to the rows here, and there are two conditions, control condition versus an experimental perturbation. And a standard differential expression analysis looks at each gene in isolation and just produces a t-test p-value or a fold change. And the problem is that it often misses the forest for the trees because it doesn't recognize that thousands of genes can be highly correlated with each other. And so the philosophy underlying weighted gene co-expression network analysis is to really focus on higher order terms. So it's a systems biologic approach where we focus on gene modules as opposed to individual genes. So this focus on gene modules also has the great benefit that it alleviates the multiple testing problem. So instead of relating every single gene or each of the 50,000 transcripts to disease status, we may relate as few as 50 modules to disease status. So to give you an intuition, then a module is a cluster of genes, just to tie it in with the previous lectures. And here we use network terminology because it's very intuitive to biologists. And all of us function, of course, in social networks, and it turns out we have a tremendous intuition for understanding network statistics. So what is weighted gene co-expression network analysis? So it typically proceeds along these following steps. So we start out constructing a network, for example, between genes, and the rationale is that we want to make use of the interaction patterns between the genes. Why? Well, genes often function as part of pathways and so on. They interact. In step number 2, we then identify gene modules, for example, using hierarchical clustering. And the rationale here is that we want to carry out a module-based analysis. And in step number 3, we relate these modules to external information because we want to find biologically interesting modules. So what is external information? It could be clinical data, for example, disease status or survival time. Or we could relate modules to gene ontology information, just to ask, is my module enriched with cell proliferation related genes? After this step, we hopefully have found an exciting module. And then we come to the next step where we want to study module preservation across data sets. For example, to check the robustness of your module, you want to see whether it can also be found in an independent data set. Or you could have a module found in human brains, and you want to know, well, is this module also preserved in chimpanzee brains, you know? And so this is often a crucial step in the analysis. And so later today, I will teach you statistics for carrying out this module preservation analysis. But more generally, these techniques also lend themselves for studying cluster preservation. All right, after these steps, you hopefully have a robustly defined, interesting module. And then in the final step, we look for the key drivers of these interesting modules. For example, are there certain transcription factors that seem to regulate the genes? Or which genes are intramodular hub genes? Or you can do causal analyses. And the rationale here is, of course, to carry out experimental validation experiments or possibly develop therapeutics and so on. So more generally, we call these approaches weighted correlation networks instead of co-expression networks. And why is that? Because they don't just apply to gene expression data, they may also apply to methylation data or fMRI data and so on. And so these weighted correlation networks are valuable for a biologically meaningful reduction of high-dimensional data and also for the integration of what I call multi-scale data. What do I mean by that? So it could be that you have gene expression data from multiple human tissues, blood tissue, brain tissue, liver tissue. Or you could have different data modalities. You could have genetic markers, you could have DNA methylation level, metabolite data, all measured on the same subjects. And so to integrate these data, you can use WGCNA. So these techniques have been adopted by mouse geneticists and people who study metabolic syndrome, such as diabetes and so on. But also they are widely used in the neuroscientific community, for example, to study autism disease. So let me now go through the details of how we construct a weighted gene co-expression network. Now, what do I mean by a network? A network is really specified by a symmetric matrix, which is referred to as adjacency matrix. So it's a symmetric matrix where all entries lie in the interval 0 and 1. And now for an unweighted networks, the entries are either 1 or 0, meaning the two genes are connected or not connected. But for a weighted network, the adjacency matrix reports a real number, which is the connection strength, right? And so how do we get an adjacency matrix based on gene expression data, which could be array data or RNA-seq data? Well, the first thing we need to do is we need to measure co-expression of genes. And we typically do that with a correlation coefficient. So just to be clear on what do I mean by co-expression? What does that really mean? Assume there are 100 cancer patients. And so here 100 arrays on the x-axis. On the y-axis shows you gene expression levels of different genes. And notice there are two genes colored in green where the two genes sometimes have a small value in some subjects. In other subjects, they have a high value. So these two genes are either both highly overexpressed in one person or they are underexpressed in the next person, and so on. They go up and down together. And that's called, they are co-expressed across human subjects. Now, to measure co-expression of two genes, you can simply correlate their gene expression levels across 100 subjects, for example. And so what you end up getting is a big matrix, which could be 50,000 times 50,000, corresponding to the transcripts, and the entries of that matrix report, for example, a correlation coefficient, measuring co-expression. And then what you need to do is you need to transform this matrix into an adjacency matrix. Why is the correlation matrix not a network? Well, the answer is because it has entries that can be negative, right? The correlation coefficient takes on values between -1 and +1. And an adjacency matrix takes on values only between 0 and 1. So you need to transform it in some shape or form. One way to turn a correlation matrix into an adjacency matrix is simply to threshold it. You say if the correlation is bigger than 0.8, I connect two genes, and if it's less than 0.8, I don't. Then the adjacency is 0. But in the weighted network analysis, we take what I call the holistic view. We assume everything is connected, but what differs is the strength of the connections. You know, there are some genes that are highly connected, indicated by a thick line, and some genes that have only a negligible connection, indicated by a thin line. In unweighted networks, you only ever have an edge or you don't have an edge. And so now the problem with the unweighted networks is that they lead to a loss of information. For example, imagine you have a threshold of 0.8, where you say if the correlation is bigger than 0.8, I connect the genes. And what happens if a pair of genes has a correlation of 0.79? You know, you will say, well, they are not connected. But you feel this isn't quite right, you know. Why? Because the data are noisy, you know, and you could say, well, if I only had measured one more microarray, then it would have been above the threshold of 0.8, you know. And so therefore, we prefer to really keep this continuous co-expression information without hard thresholding it. And so we construct two types of weighted correlation networks. The first type is called unsigned network, and the second is called a signed network. Both of these networks are defined with respect to a correlation coefficient. So let's start with the first network. So we take the correlation between two genes, I and J, and then in the unsigned case, we take the absolute value of that correlation coefficient, which results in a number that lies between 0 and 1. Now, if you take such a number and raise it to a power, it's again is a number between 0 and 1, you know. And so the right-hand side really defines then an adjacency, right? Because the result is a number between 0 and 1. Why do we call it unsigned? Well, because it doesn't distinguish between a positive correlation coefficient and a negative correlation coefficient. Right, a correlation of 0.8 gives you the same adjacency as a correlation of minus 0.8. It ignores the sign information. It's unsigned. In contrast, a signed network is arrived at as follows, which looks a little bit weird, but the main purpose of this transformation is to arrive at a number between 0 and 1. And then again, you use a power. Why do we use a power, by the way? What's the purpose? The power is a threshold. It's called a soft threshold. It's meant to emphasize large correlation at the expense of low correlations. You know? So these powers, which we call beta, have certain default values that work very well in practice. So we use a power beta of 6 for an unsigned network and a power beta equal to 12 for a signed network. And just to tell you, in recent publications, we only ever use signed networks. Why? Because the modules that we find turn out to be more biologically more meaningful. You know? Remember, the name of the game is to find interesting modules. That's what you want. Yeah, please.
How do you preserve the sign if you still have an absolute value to
Yeah, actually, you don't need to form the absolute value here because this maps a number into the unit interval. So it could be a round bracket, but but let me. That's a very good question. How do we preserve the sign? So let's move to this slide. So here on the right panel, I show how the correlation on the x-axis relates to the adjacency on the y-axis for different threshold choices. Each line corresponds to different power. Now, notice when you have reasonably large powers, then all the negative correlations always result in an adjacency that is very close to 0, right? What does that mean? Genes that have a negative correlation with each other are in essence not connected in the network. You know? Only genes with a positive correlation get connected. You know? In this sense, the signed network distinguishes positive from negative correlations. Contrast that with the unsigned network on the left panel. So what you see is you get these U-shaped curves. Why? Because a negative value gets the same adjacency as a positive value. You know? So the other thing you notice is the higher the power, the more emphasis is placed on positive or high positive correlations, right? If you have a high power, then a correlation of, let's say, 0.1, 0.2 gets pushed towards 0. Only very high positive correlations result in non-negligible adjacencies. Yeah. Yes? Yes. Automatically, because look, if you have a correlation minus 0.5, pretty much any power will set the adjacency to 0. And so, what does it all mean? It means when we later get modules, then modules in a signed network are comprised of genes that all have positive correlations with each other. Whereas in an unsigned network, you may get modules where the genes have strong negative correlations with each other. You know? That's a basic distinction. And there's really no right or wrong answer. There will be some applications where you feel that I'm very interested in modules that may involve negatively related genes. Then go with that method, you know. But I'm just telling you, in 90% of cases nowadays, we use signed networks. You know. So this is question 1. Should the network construction account for the sign of the co-expression relationship? And so here's one article where we applied these methods to embryonic stem cells, and it was critical to use a signed network. Another question is, why construct a co-expression network based on the correlation coefficient, right? Because you can measure co-expression with many other measures, mutual information, there are many dependence measures. And well, the reason why we use it manifold, there are many reasons. One is, it's of course intuitive. Correlation measures is a widely used measure of relationship. Pearson correlation measures linear relationships and avoids the pitfalls of overfitting. But you may know there are many correlation measures. There's something called the Spearman correlation, which measures monotonic relationships. And there's another correlation measure that probably you don't know, which is known as bi-weight mid-correlation. It's another robust correlation coefficient. And it turns out this by-weight mid-correlation seems to be the best measure. We evaluated them. And so later I'll show you how to get it. In any event, coming back to why do we like correlations, they work very well in practice, computationally fast and reproducible research. And they are particularly recommended if you have relatively few arrays. I want to say if you have fewer than 50 arrays, I urge you to use correlation and nothing more complicated. You know. Why? Because a lot can go wrong when you estimate nonlinear relationships. For example, some outliers may mislead you to think that there's a nonlinear relationship. So Lin Song has carried out a very comprehensive comparison of different co-expression measures. For example, mutual information versus correlation coefficients and many other measures. And so I want to recommend to you, if you are interested in that topic, to look at this recent article where she looked at 8 different data sets. It was a very long and comprehensive comparison. But what we found was that this by-weight mid-correlation that I mentioned to you, coupled with another measure called topologic overlap, worked best at defining co-expression modules that, what do I mean by working best? What is the criterion? The criterion was enrichment with gene ontology categories. You know, you can ask, if I find a cluster of genes, is it enriched with certain Go categories? And so you can then evaluate different procedures for arriving at these modules. All right. Why soft thresholding as opposed to hard thresholding? Well, soft thresholding preserves the continuous information of the co-expression, right? We don't threshold it at 0.8. We keep it continuous. And also, there's strong empirical evidence that the resulting networks are more robust with regard to threshold choices. So if you use a soft threshold of 6 or a soft threshold of 8 or 10, you get very similar modules. But if you use a hard threshold, let's say 0.5 versus 0.8, you get very different, it's less robust. But there is one major use of hard thresholding, meaning unweighted networks, which is you can use graph theoretic algorithms from the computer science community to analyze your data. All right, coming to the advantages of soft thresholding. I mentioned robustness. Now I want to highlight a few other reasons. Another major advantage of weighted networks is that they allow you to calibrate the networks. What do I mean by that? Imagine you have 8 different lung cancer data sets from different labs measured on different array platforms. And you want to combine the data. So one way to do it is to build a network for each individual data set. Each lung cancer data set gives you one network. And so you end up with 8 different networks. But now what you would like to do is you want to combine these 8 networks into one overall lung cancer network. And later I teach you techniques how to do it. But it turns out that a critical step is to calibrate the network so that they become comparable. Why? Because they differ. Some networks, some data sets involved the Agilent technology, others the Affymetrix technology, others are RNA-seq. They may be comprised of different ethnic groups. One is Japanese, the other is Caucasians, and also different sample sizes. And so a critical step is to quote-unquote calibrate or normalize the network so that they become similar. And that normalization or calibration step is very straightforward when you deal with a weighted network. Why? Because you have real numbers. You can relate them so that the distributions are comparable. For example, you can say, I want the maximum adjacency to be the same, or the median adjacency to be the same. And so that's straightforward. Another reason why we like these weighted networks is that you can develop module preservation statistics that are particularly sensitive. Remember how I said one critical step in network analysis is to assess whether your favorite module is reproducible or preserved in an independent data set. And to do that, you need module preservation statistics. And so, again, weighted networks give rise to very powerful statistics. And then the other reason why we like the power function, meaning we raise the correlation to a power, there is a mathematical reason for it, and I urge you to read the papers if you want to learn more about that. All right. Do you have any questions at this point? Okay. So one question one can address is, how do we choose this power beta? Remember the threshold that I mentioned earlier. This threshold. How do we choose it? The power. Well, we do that by looking at the distribution of connectivities. And let me explain that to you. So I first need to explain what do I mean by gene connectivity. Remember, you have an adjacency matrix, and each row corresponds to a gene. So you can just sum up all the adjacencies of that gene, and that is called the ith connectivity, for the connectivity of gene I. And in a social network, that is simply the number of friends somebody has, the number of neighbors. Now, what one can do is one can plot a frequency plot of the connectivities, which is done here, also known as a histogram. And so the x-axis shows the connectivity values, and the y-axis shows their frequency. And notice we arrive here at a highly skewed distribution, meaning most genes have a low connectivity. Or in a social network, most people have no friends, But there's some people or some genes that have a lot of connections. These genes with a lot of connections are known as hub genes, highly connected hubs. So in any event, it turns out that observing such a distribution of the connectivities is desirable for various reasons I mentioned. And as a matter of fact, Dr. Barabasi noted this property first, and as he called, just very roughly speaking, he referred to this highly skewed distribution as scale-free topology. It's a fancy name. But let me give you a more precise definition. What is a scale-free network? So instead, remember, here's connectivity, here's the frequency of the connectivity. Now let's plot the same graph on the log scale, where we show the logarithm of the connectivity versus the logarithm of the frequency. Then it turns out often you observe a straight line relationship. And if you do observe a straight line relationship, then one says the network is scale-free. And so there's a vast literature on scale-free network. Why are they good? Why are they bad? And as I mentioned, so for example, Dr. Barabasi's group has investigated these properties of these networks. Now, for our purposes, we use this type of behavior to choose a power. And remember, this is under the subject heading, how do you choose a threshold? just to give you the answer already, I choose the threshold so that I get a scale-free network. That's the, the answer, you know. So instead of writing papers where we say, in the mouse liver data, we find a scale-free network, we turn things around. We say, we chose the power so that we will have a scale-free network in the data. Do you see? Different logic. Not circular logic, but different logic. So in any event, to flesh this out, let me give you another terminology. Note, as I said, a scale-free network is characterized by a linear relationship here. And how do you measure linear relationships? Well, you can again just use the correlation and square it. This is the R-square measure. This correlation between the x-axis and the y-axis squared is a very good measure for assessing a scale-free topology. And just to give you already the heads up, so what we do is we plot this R-square measure on the y-axis versus different powers for thresholding and see how the R-square measure changes, you know. And so, so do you see, when you have a low power, you don't have a scale-free network. The R-square is close to 0. Only if you observe a high power, only then do you get a scale-free network. And so this insight then can be used to say, well, I choose the power, for example, here 6 or 7, so that I get a scale-free network, you know. That's the essence of the criterion. Yes?
If you, for example, say it stays on 0.6, then it's not pretty much a scale-free network, or
Yeah. Well, okay, just to so the way I use the criterion, I don't so much look at the axis, at the absolute value. I often observe this point where it goes up and then it levels off. And I use the lowest power where I get this saturation point, you know.
and you always get the saturation point?
No, not always. Everything can go wrong. So, which comes back to another question before we get there. Why do we like scale-free topology? It's actually a controversial topic. Many biologists roll their eyes and say, "Oh, come on," you know. So, and why? So the thing is that when you deal with microarray data from homogeneous, from a single tissue, let's say mouse liver expression data, then we usually find that relatively low powers allow us to achieve scale-free topology. And in this sense, scale-free topology almost serves as a data quality metric. If the reverse situation happens, if I don't find a low power that gives me scale-free topology, I start to become nervous. I say there may be something wrong with the data. There could be artifacts. So if you have severe technical problems, severe batch effects, you don't get scale-free topology. But there could also be a biologic reason why you don't get scale-free topology. Assume, for example, that you have two types of tissues. You have muscle tissues and you have liver tissue, different tissues. And you now want to construct a co-expression network. Chances are you will not get scale-free topology. And why is that? Because in essence, you have one big module in the data. You know, there's a module comprised of genes that are overexpressed in liver tissue, and actually there's another module comprised of genes that are overexpressed in muscle tissue. Two gigantic modules. And then, no matter how you choose a threshold, you don't get scale-free topology. You know? So in certain ways, the scale-free topology measures a bit the module structure that you have. If and also it measures Well, coming back to this, maybe that's a better way to look at scale-free topology. So often we want that most genes have a low connectivity. Why? Because or a substantial number of genes don't do anything. They shouldn't interact. And so then you would like to see this type of a histogram. But it does happen that sometimes you don't get this histogram. You actually get that most genes are hub genes. Everything is connected. And then there may be a good biologic reason why everything is a hub. Again, that could be because you have two different tissues. They're all hubs because they're all part of the muscle genes or they're part of the liver genes, you know. And so then you have a good reason not to see it. But in many cases, you want to see that type of histogram. You know. All right. Now, as I mentioned, Barabasi had very different reasons for wanting scale-free topology, robustness of the network, and so on. But I don't mention them here because that's in the realm of co-expression networks. These original reasons may not be pertinent. You know. Yeah. Other questions? If you think this is too complicated, just go with the default choices. I often do. I don't even think about it. I choose the power of 12 for a signed network and never mind the rest. Okay. And here's an example of a yeast network. And you notice there's this quadratic relationship. So there are definitely co-expression networks where you don't observe scale-free topology. So remember, scale-free topology means a straight line relationship. All right, so let's move to another exciting topic, which is how do we measure interconnectedness in a network? You know? Coming back to a social network. Let's say we have a social network and the adjacency matrix specifies how much two people like each other. Then, of course, one way to measure connections is by the adjacency matrix. It just, as the name indicates, it measures how adjacent two things are. But there's a second measure, which is known as topologic overlap measure, or matrix. And this measure has actually a very nice intuitive explanation. So, coming to the social network, it measures the proportions of friends you share in common, okay? So let's say you have two people, instead of asking, are the two of them friends, which is a non-robust measure, you know, because people may be friends today and may not be friends tomorrow, you know, it's not robust. However, what is much more robust is to ask, well, do they share the same friends, you know? Because if two people share a lot of friends together, chances are that after a week they make up again and then they are friends again, you know. and so that intuition is accomplished by the topologic overlap. And so mathematically, the topologic overlap measure is defined using the matrix product of the adjacency matrix with itself divided by the minimum of the respective connectivities of the two people. And you can show that this measure takes a value between 0 and 1. So the topologic overlap measures similarity, and to turn it into a dissimilarity, we take 1 minus it. Why do we turn it into a dissimilarity? Well, because we use it as input of hierarchical clustering. So we need a dissimilarity measure. Let us now come to the topic of how do we detect network modules, meaning clusters. Well, as I mentioned, we use average linkage hierarchical clustering coupled with this topologic overlap dissimilarity measure. And then we get a cluster tree and we define the modules as branches. For example, here's a cluster tree that I showed earlier, and then the modules correspond to these different branches. And it is very tricky to cut the branches. And so there's an R package, which I will teach to you shortly, called the dynamic tree cut approach, which accomplishes this branch cutting for you. So here's the R code, cut tree dynamic, cut these branches in an adaptive fashion. So now I want to illustrate this method using a toy example, where again the individual objects correspond to integers, 1, 2, 3, 4, and so on. And I use hierarchical clustering to cluster them. And look at the tree. A question is, well, how many clusters are there, you know? You can look at it in a variety of ways, but one way to look at it is this one is a branch comprised of the numbers 1, 2, 3, 4, and 5, the left side. This is another branch, and this is a third branch. One way to look at it is there are 3 clusters in the data. And now, again, the dissimilarity between 2 numbers is the absolute value of their difference. That's how that was the input. There's one number, the number 54, that is an outlier. It's very far away from all the other numbers. Do you notice? Yeah. Which tells you already how an advantage of hierarchical clustering. It can deal with singletons, you know. It doesn't force 54 into this cluster. It just leaves it where it is. Okay, so how would one then arrive at clusters? Well, one way is to cut it in a static fashion. Do you see the red line corresponds to a height cutoff? Remember, the y-axis is the distance. And so you fix a static cut and you say any branch underneath this red line gets a cluster label. So there's the turquoise, the blue, and the yellow cluster. The other thing is, you notice the color gray. That is reserved for objects that are not part of any cluster, right? Which is a major strength of hierarchical clustering, because you get objects that are not forced into a cluster. But in any event, looking at this dendrogram, there's one thing unsatisfactory, which is you feel, well, why wasn't 19 and 38 grouped into the yellow cluster, you know? They are part of this branch. Well, the problem is that you have one fixed height. What you need really is, well, one way what you could change is you could say, well, I raise the height, you know, and choose another height cutoff, and now I have 2 branches. Now I get this branch right, the blue cluster, but I merged these 2 clusters. I'm not happy with that. So what you really want is a dynamic cutting approach that forms one cutoff here, one here, and one here, meaning every single branch gets its own height cutoff, an adaptive approach. And that is really accomplished by this dynamic approach, you know. So in summary, these are all the cluster assignments. This is for a constant threshold of 6.5, a constant height threshold of 15, and this is the dynamic hybrid method. And so here in this toy example, the hybrid method really adaptively chooses the perfect height for each branch. Let's come to a more complicated situation. Again, simulated data. I mean, just looking at it as a human being, how many branches do you see? Here's 1, here's 2, and here's 3, you know. Notice the great advantage of hierarchical clustering. This simulation involves maybe thousands of genes. How would, if you didn't see the tree, you may be tempted to say, well, wait, there are 1,000 genes, I better think there are at least 10 clusters, you know. But just looking at the tree, you say, no, there aren't. It's about 3, you know. And the other thing you notice on the leftmost side, you see a lot of genes that are at the top of the tree. They are singletons. They're really not part of any cluster, you know. In any event, because we simulated the data, we know the true cluster assignment. And so what do we learn? Well, there's the turquoise branch, and then there's the brown branch, and the blue branch. But something interesting is going on. Notice that a lot of the turquoise genes end up actually also here, do you see, to the left of the brown branch. In any event, so the approaches don't work. In contrast, when you use the dynamic approach, it already captures the true underlying cluster structure much better. But here we distinguish between two types of dynamic branch cutting approaches. There's the first approach called dynamic tree method, and the second approach called dynamic hybrid method. And as I indicated a couple of times, we really like the hybrid method. But for historic reasons, let me explain the tree method. The tree method is called tree method because it takes as input nothing but a tree. And so you give it this cluster tree, and then the method adaptively chooses the optimum height cutoff for each branch using a certain criterion. So it works quite well. But but the problem here is, notice that it assigns the color brown to all of these turquoise genes, you know. It overdoes it. So in contrast, the dynamic hybrid method really captures the underlying true signal. And how does it do it? Why does it work so well? Well, because the hybrid method uses a second clustering procedure after the tree approach, which is based on partitioning around medoids. The term hybrid refers to the fact that it's a hybrid between hierarchical clustering and partitioning around medoids. And remember how I told you the hybrid method inherits the advantages of both methods. It doesn't require you to specify the number of clusters, but it's also much more robust because it reshuffles the cluster assignment. The hybrid method says, let me look at the cluster assignment one more time, and I will test every single gene whether it shouldn't be part of the other branch. It allows this reshuffling. And so therefore, I mean, the drawback is that you get these non-contiguous colors, you know. It looks a little bit messy, but it is much more robust. So, yeah, let me mention then that this hybrid method, as I mentioned, is more flexible. It can deal with very complicated dendrograms, very good at outlier detection. It's suitable for automation, and it's much more robust, you know. And so I really recommend it to you as a clustering procedure. The dynamic tree cut method doesn't require that you use the topologic overlap measure or any other measure. You can use it for any clustering assignment. You can have your favorite distance or dissimilarity measure, and you can use it as input of this algorithm, you know. Any questions at this point? Okay. Yes?
General question. Biologically, one can think of examples of, say, transcription factors that are expressed at a relatively low level, but upon binding of a ligand, for example, and induce the expression of a number of genes. And those two would be very closely related biologically, but if the expression of the transcription factor itself is not increased, but
yes.
changes the topology, physiologic change in it that leads to something else, it wouldn't be captured as part of the same.
Yes. Yeah, that's a very good criticism. So or comment. Because I'm on video, I will repeat it. So the comment was that transcription factors may have expression levels, mRNA levels, that really don't relate to the mRNA levels of the target genes, you know, because it happens at the level of proteins. And we observe that, often transcription factors really are not co-expressed with the target genes. And so yes, so in this case, co-expression really doesn't capture it, And so you would therefore want to use a different way of constructing a network adjacency, It could, you could think of defining an adjacency that measures occupancy somehow between genes and transcription factors, And so, yeah, that's why I gave a very general definition of what is an adjacency matrix. It's a symmetric matrix, entries between 0 and 1. And so the biologists can define it any way they want. The good news for you is the WGCNA package really allows you to analyze any adjacency matrix. We used it for protein-protein interaction data, whole range of data. The topologic overlap measure is sometimes very useful when you deal with sparse networks, meaning a lot of zeros. For example, in protein-protein interaction networks, there's often little information whether two proteins are connected or not. And again, there several authors, Horvath et al. And so on, they showed that using the topologic overlap leads to better results. Yeah. Other questions? Good. But on a more deeper level, what is co-expression? When people first proposed these methods, maybe they were hoping that it would reflect signaling pathways, And this is largely, that's not true, And when you have heterogeneous tissues, then co-expression has often a weak relationship with protein-protein interaction, That could be different when you have single cell analyses, But basically when you have heterogeneous tissues, then modules sometimes correspond to cell types. That's certainly the case in brain tissue. If you take human cortex and you form a co-expression network, then you will find modules that correspond to different cell types, oligodendrocytes, neurons, and so on. So co-expression is not the same as a signaling relationship. All right. How do we visualize networks? There's a variety of tools. One is called the topologic overlap matrix plots and so on. I just briefly review it. Why? Because often you see these plots in scientific papers and so. So there's something called the topologic overlap matrix plot, which is the following. So here, this matrix visualizes this distance measure, the topologic overlap between genes. And what you see is the rows and the columns correspond, of course, to the genes, but now they are sorted according to the hierarchical cluster tree, And so, so the modules then correspond to squares along the diagonal where you see a thick, a dark yellow. What does dark yellow means? Highly connected. So for example, here the turquoise branch corresponds to this square, and you notice that the genes are highly connected. Being a biostatistician, I don't like this plot because it doesn't tell me anything. For a statistician, the only thing that matters is really the dendrogram, right? The dendrogram is used as input of branch cutting for defining clusters or modules, whereas the lower part is just a colorful, All right, now another way to visualize a network is via something called multidimensional scaling. So that is a projection of the network into, for example, the two-dimensional plane or into a three-dimensional space. Each dot corresponds to a gene, and you notice that, so I color, of course, the genes according to their module membership. And what do I get? Essentially, I get a hand with fingers on it, And the fingers correspond to the different modules. And what we observe is that these the gray dots, remember gray are genes that are not part of any module, and they lie in the middle. Why? Because they don't know which finger they belong to. They are confused, you know. The other thing you can ask, so what's special about genes at the tips of these fingers, right? They seem to be special. And it turns out that these genes in the tips are what we call intramodular hub genes. They're hubs. They're centrally located in the module. A traditional view of a network is, of course, provided by this type of plot where you have dots and you just connect them if they are connected, you know. And finally, I mentioned that, of course, what we do with modules is we study their gene ontology enrichment. So using any number of tools, for example, the David tool, do you know this? Ease, David, Ingenuity Analysis, Web Gestalt, there's a lot of wonderful software packages out there. And so, for example, we would say this brown module here is highly enriched with mitosis-related genes, you know. And so we would learn something about the biologic meaning of the modules. Here is a heat map view of a module. And what we observe, so what do we see here? So the rows correspond to genes, and the columns correspond to different tissue samples. For example, 100 different cancer subjects. You know? And what we observe is these vertical bands, right? Why do we observe it? Well, the vertical black indicates the genes are highly overexpressed. And so what we learn is the blue module genes are highly overexpressed in these people, but they're underexpressed in these people. Green means underexpressed. And so the vertical bands indicate that these genes are co-expressed. They go up together, they go down together. And so, so it's no surprise, these are co-expression modules. You know? How do we summarize the expression profiles of a module? Towards that end, there are really two answers. One is to form a weighted average or principal component, and the other answer is to use the most highly connected hub gene in the module. And both turn out to be equivalent. So let me explain this a little bit more. It's actually a very important concept. What is a module eigengene? It's really essential to everything we will do. So let me start out by studying one particular module, the brown module. So the rows of the heat map correspond to genes. The columns are different human subjects. And what we observe is, for example, there's one microarray where all the genes take on a red value, meaning they're all overexpressed in this human being. And in these human beings, they're all green, they're underexpressed. And so the module eigengene is really a weighted average of these genes. And here, the bar plot shows the values of the module eigengene across these different subjects. For example, for this person, where all genes are overexpressed, the module eigengene takes a positive value. It measures overexpression. For these subjects, where all the genes are underexpressed, the module eigengene takes negative values, right? And so in this sense, the module eigengene really captures the pattern that you see here, you know. It is the mathematically optimal way of summarizing the gene expression values of the module gene. You know. For the experts who like that type of stuff, I mentioned that mathematically we define the module eigengene using the singular value decomposition. So we scale or standardize the gene expression data so that they all have mean 0, variance 1, and then we do a singular value decomposition and define it as the singular vector. the word eigen reflects something like eigenvalue, eigenvector, you know, that's where it all comes from. If you've learned this in linear algebra, I don't require that you know what it means, just so that you know. Okay, so why are module eigengenes very useful? Well, they allow us to relate modules to each other. So for example, they allow us to determine whether modules should be merged. Assume you have the blue module and the brown module. Each module gets summarized by its eigengene. Then you can correlate these eigengenes and say, oh wow, they have a correlation of 0.9. They're highly related. These modules are very similar. And then you say, I will just merge the two modules, you know. That's how you could use that. The other way to use modules is to relate them to clinical traits. So for example, let's say you have 50 modules, then you have 50 module eigengenes. And so now you relate each module eigengene to disease status. And you find, wow, one of them relates highly to the disease. And by focusing on module eigengenes, you really avoid the multiple comparison problem. Instead of relating 50,000 transcripts to disease status, you relate 50 module eigengenes, you know. They also, apart from relating modules to disease states and avoiding the multiple comparison problem, we can module eigengenes to define a measure of module membership. Namely, this can be accomplished by correlating each individual gene with the module eigengene, you know. So assume you have a module that is very interesting to you, the brown module. And you take its eigengene and you correlate it with each of the 50,000 probes or transcripts that you study. And so then you get this correlation coefficient called KME, and you just say, well, what is, you can rank order the genes, and you say, give me the 500 genes with the highest correlations with the module eigengene. And then you take these 500 genes and you use them as input of a gene ontology enrichment analysis, you know. And you say, oh, they are all highly enriched with mitosis-related genes, you know. So this measure is called connectivity based on the module eigengene and often referred to as KME. But it is a module membership measure. Yes?
So I'm not sure I understand that. You're talking about doing that on the same expression data or
yeah, same expression data. Mm-hmm.
But you already defined the membership in that data, so why would you think there'd be other genes that would also be correlated? Wouldn't they have been part of the module in the first place?
Roughly speaking, you're right. However, it turns out, of course, that often the user made certain choices. Let's say the branch cutting. Let's say you got it wrong, you know, and you missed some genes. Or in the data pre-processing, somebody said, I don't analyze all 50,000 genes together. I just form modules based on 10,000 genes that are highly expressed in brain tissue, you know. So then you want to go back and relate all the genes to the module, you know. And also, this measure is attractive because it allows you to rank order genes, right? Let's say we have a branch of 500 genes. You actually don't know, well, which gene is in the tip of the branch. Sorry. Let's say here we have these branches, right? Now, if you say, oh, I just take all these blue genes, you really don't know, well, which genes are in the tip and which are higher up. The KME measure, or intramodular connectivity, allows you to say, well, these are the hubs. It turns out the genes, measures the genes with high intramodular connectivity or high KME, they are in the tip of the branch, you know. And so the KME measure is a quantitative measure of module, you know. And it also has this other great advantage that it allows you to study how a gene relates to multiple modules. So it could be, of course, that you have a gene that has high KME with the blue module, high correlation with its eigengene, but it also has a high KME with the brown module. There could be genes that are part of both modules. The KME measure allows you to retrieve that information, you know. So that's, so the KME measure is really then a quantitative measure of module membership. Yeah. But it's also used to find hub genes, as I mentioned. Yeah. So, yes?
This question will make sense, but a problem we have with RNA-seq data, for example, is that it's really difficult to make comparisons of gene expression within a sample, right? You can compare the same gene across samples, but within a sample, this gene versus that gene is a problem. Does that also apply to modules, for example? Or are these comparisons always across?
Yeah, so everything here is fundamentally co-expression. It always requires that you have multiple arrays, or not arrays, it could be subjects, you know, tissue samples. So I want to say about, you want at least 20 samples, you know.
The comparisons are always made across?
Yes, it's always about co-expression. It's not within a sample, yeah.
So you have,
Yeah.
so you have a module eigengene value per module per sample.
Yes,
And then you compare those across samples.
yes, yes.
You just said example to the top 1,000 gene from the brain tissue.
Yes.
How do you decide the top 1,000 genes?
No, not 1,000. I said 10,000, but yeah, it's a slight variation, but I would. So it sometimes you have, of course, gene expression data from multiple tissues, blood, brain, especially in when you deal with mice. So people have multiple tissues from mice. And then you could say, you know, I really want to study brain function. So I want to find genes that are really highly overexpressed in brain tissue. And then you would compare it with some sort of a t-test. You say which genes are high in brain tissue compared to blood and muscle tissue, you know. And then you could say, well, I just take the top 10,000, you know, that are overexpressed in the brain, you know. No, yeah, you need a comparison tissue, absolutely, you know. You know, these tools, I mean, personally, I always apply it to all data. Why? I'm too lazy to sort genes. I apply to all 50,000 or whatever transcripts, you know. But I have colleagues who are neuroscientists, and they really know what they are doing, and they often do a filtering step. The idea is you remove noise in some shape or form, you know. All right, let me come to one application so that you get a sense of what this looks like. So here, my colleagues, Chris Plaisir and Päivi Pajukante, studied hyperlipidemia in human beings, and they had actually adipose tissue samples from 75 human subjects and measured gene expression data. And then what they did is they found these modules indicated in the rows, and each module is summarized by its module eigengene. So for example, the turquoise module eigengene corresponds to a module that contains 1,600 genes. You know, these numbers indicate number of genes. And the columns correspond to different clinical traits: familial combined hyperlipidemia status, total cholesterol, triglyceride levels, ApoB levels, and there's one particular SNP that they were interested in. Now, the entries of the table report correlations and, in brackets, a p-value. For example, there's this one module indicated by the color tan in the middle whose module eigengene correlated highly with hyperlipidemia status, also with total cholesterol, also with triglyceride levels and ApoB levels, and also with the SNP, you know. So this is a very exciting module, you know, biologically speaking, if you study hyperlipidemia. By the way, why did they study this one SNP? Well, it was found in a linkage analysis of some Finnish pedigrees. This was a SNP that was very interesting to them. And so it's very interesting then that the SNP relates to this module in human fat tissue. There's clearly something going on. But now let's look at, well, let's start looking at the correlations. So what do we see? A correlation 0.4, correlation 0.3, correlation 0.3. What do you think? These are high correlations, you know. If you study a complex trait such as hyperlipidemia, that's a high correlation. Now look at the p-values though. They are in principle 10 to the minus 4, you know. I mean, nothing to write home about if you correct for 50,000 comparisons. If you do a multiple comparison correction for 50,000 tests, that none of these results would be significant. However, we don't have 50,000 comparisons. We have here 28 modules. And if you do a Bonferroni correction for 28 comparisons, that will still be highly significant, which is why it could be published, you know. And so that highlights a major utility of these methods. You reduce the data drastically, and therefore you manage to find significant results, you know. So why did we need to do it? Because we only had 70 or so human subjects, you know. There's not many people who volunteer to give out a fat biopsy, you know. And so so the sample size was restricted, yeah. Yes?
So just make sure I understand, so this is the gene expression profile where for every patient you had a value for CSHL.
Yes. And therefore, so what I can do is I can correlate the module eigengene with disease status across 70 people. So it's a correlation value and a correlation test p-value. And you know, in a few minutes, I will give you R code that shows you how to create this type of output. It's often used in publications, you know. Why? Because you may have your favorite disease that involves many different phenotypes. You could have 50 phenotypes, you know, and you can correlate then all of your clinical phenotypes to the gene expression data. So this type of table summarizes your entire data set in one big table, you know. All right, so module eigengenes, as I mentioned, can be used to decide whether two modules should be merged. So you can create a scatter plot, for example, here between the brown module eigengene and the blue module eigengene, and just study how are they related, you know. And so if two module eigengenes are highly correlated, then maybe you should merge the two modules. Now, let me come to another important topic, which is module detection in very large data sets. What is a large data set? For example, 500,000 variables, you know. And where do we encounter it? For example, when you do DNA methylation studies, you may know that there's DNA methylation arrays from Illumina measure 480,000 locations in the human genome. And again, you would like to then find modules based on methylation levels. And standard clustering procedures really struggle with 500,000 variables. And but fortunately, we have an R function called blockwise modules that runs on your laptop and can analyze very large data sets. And so this function proceeds along 3 steps. Assume you have your 500,000 variables. In step number 1, it would cluster these into 50 blocks where each block contains 10,000 variables, right? And so, that's where the name comes from, blockwise. So it splits it into 50 blocks of 10,000 variables each. And then it uses hierarchical clustering in each block separately. And so you end up with 50 hierarchical cluster trees. And then what it does is, we of course use branch cutting and so on, and we get in each block clusters and modules. Then we summarize each module by its module eigengene, and then we merge the results across blocks by forming correlations between module eigengenes, right? So for example, if the blue module in block 1 is highly correlated with the brown module in block 2, then we merge the two colors, you know. And so again, this is a very powerful approach that works on hundreds of thousands of variables, you know. And so I'll give you some code for that later on. How do we relate modules to external data? So let's say we have a clinical trait, for example, body mass index. One way to do it is, well, you correlate each individual gene with this clinical trait and you form the absolute value. And that is what we call a gene significance measure. Why? Because it measures how significant the gene is for body mass index, you know. But sometimes you could also have a t-test, for example, from some sort of multivariable regression model. So you could just take the absolute value of the t-test statistic. Or if you happen to have a p-value from, say, a Cox regression model, you take minus log of a p-value. So in general, these types of measures are called gene significance measures. And so there's actually only one convention. The higher the value, the more significant, you know. If you have a SNP, you can use an additive marker coding where you count the number of A alleles, for example, 2, 1, and 0. And then this additive or numeric coding can be correlated with each gene. So you get a gene significance measure based on a SNP. And then to define a module significance measure, you just define it as the mean gene significance per module, you know. Why do we do that? Why do we need module significance measures? Well, because we want to find the interesting modules, right? We want to find modules that relate to your clinical trait, for example, or modules that are enriched with certain significant genes that we care about. Let's come to another task in network analysis, which is if we have found, for example, a network of interesting and interacting genes, how do we find the hubs? And to give you a social network analogy, I show here a co-authorship network where each entry corresponds to a mathematician, and they are linked if they co-authored a math paper together. And if I ask you, well, who is the most influential author here? You probably see there's this guy Erdos. And so he's actually a famous mathematician because he published, I want to say, 1,400 papers or so in his lifetime, a vast number of, and all of them are actually very high quality papers. And so the point is that sometimes these highly connected nodes are very important, certainly in this authorship network. Here's another analogy from Albert Barabasi's. So this map shows flight connections for a particular airline. And what you notice, there are certain cities that are hubs, you know, that connect to a lot of other cities. And so again, here that indicates that nodes with high connectivity are very important. And so this type of intuition led people to suspect that hubs in gene networks are also important, right? It's very tempting to come to that conclusion. And people worked on it. And I also worked on it. But the problem is that this intuition is actually wrong, you know. Sorry for disappointing you, but when you have a network comprised of, let's say, 20,000 genes, and you look at the hubs in that network, the most highly connected genes, chances are these are completely uninteresting genes for your research study. And let me mention that the story changes when you focus on particular modules. You know, remember how I always mentioned to you a major goal of WGCNA is to find biologically interesting modules. Why? Because you then focus on the modules. See, when you have a very interesting module, then finding hub genes inside that module, that is a very fruitful enterprise, you know. So intramodular hubs can be hugely important. It's just that whole network hubs are not important as a rule. And so therefore, let's come to the definition of measures of intramodular connectivity, because they are meant to find these intramodular hubs, you know. So there are two measures. The first measure of intramodular connectivity is defined simply by the sum of adjacencies that a gene has with all the genes in that module, right? Let me give you a social network analogy. If you have a high school and then the modules in that high school are comprised of students that have shared interests. There's a football club, there's the basketball club, there's the chess club and the science club, for example. And then intramodular hubs are really the kids that are most popular within these different modules, you know. Right? It's the number of friends that you have with respect to the science club, you know. That's your intramodular connectivity. Another measure that I mentioned is this KME measure, that you can see. This measure is based on adjacencies. You can define it for any network, protein-protein interaction networks or anything. This measure you can only define for correlation networks, right? Why? Because you have a module eigengene and you correlate it with the gene expression profile. So that's the KME measure. Now it turns out that the KME measure and the intramodular connectivity are highly related. Without giving you the details, here are plots for different modules where I relate intramodular connectivity on the y-axis versus the KME measure on the x-axis, and you notice almost perfect relationships, you know. So in practice, whether you use K-in or KME in case of a correlation network doesn't matter. So you might as well use KME. Because the KME has this several advantages, which is, for example, it's naturally scaled. The KME is a correlation, and therefore it's always a number between -1 and 1. It also contains the sign information. You can say, is my gene positively related to the module or negatively related? And also, of course, you can use a p-value if you want to calculate p-values for this correlation coefficient. All right. As I mentioned, we care very much about intramodular hubs in interesting modules. For example, you could ask, if I have a cancer-related module, well, what are the hubs? And I've done that exercise several times, and often it turned out that the hubs are known cancer targets, you know. You can also do differential network analysis, where you ask, if I have a gene that is a hub in human brains, but not in chimpanzee brains, then maybe this is a gene that really reflects species differences. So that's known as differential network analysis. So how do we use networks for gene screening, to find the critical genes? Well, we usually plot the gene significance measure versus intramodular connectivity. So each panel here corresponds to a different module, and the x-axis shows intramodular connectivity and the y-axis shows a gene significance measure, for example, a t-test statistic. And what we observe is that, for example, here the brown module contains hub genes that have a high significance, t-test 4, meaning hugely significant. In contrast, so that's for the brown module. Notice for the black module, the story is very different. So the hub genes, the genes with high intramodular connectivity, actually have a low gene significance, you know, and which reflects my statement made earlier. Hub genes are not always important. It depends on context, you know. Hubs in the brown module, very exciting. Hubs in the black module, not exciting, you know. So in practice, when we use networks for finding exciting genes, we hone in on the modules that relate to that have high gene significance in one shape or another. They're biologically interesting. And then within these modules, we look at genes that are both hubs, but also have high gene significance, you know. It's this combination. Any questions at this point? Yeah. Yeah.
I guess I'm a little confused about, and I just need you to explain it again, gene significance versus, I guess I'm confused about which genes are significant versus not significant.
Oh, okay, yeah.
I guess just the statement of uninteresting versus interesting, and
yes.
an example would help.
Yes, let me start out, before we go to that example, let me start out with a social network analogy, okay? Imagine you are a faculty member in your department and you want to recruit a science students for your department to admit them. So then what you know is you know the person's grade point average, you know, and that would be the gene significance measure. So every person their significance for science, let's say, is in part reflected by their grades, you know. That's a reasonable significance measure in this context. But also what you know is for each person, you know their module membership measure with respect to different high school groups. So in particular, you know, well, this person was really popular in the science club. This was the most popular hub. Everyone knew him or her. And, well, suddenly it becomes very relevant to have both of these informations because, of course, you want to recruit people with good grades, but clearly somebody who was highly respected by their peers in the science club, chances are they are impressive people in that context, you know. And so you want to therefore use both measures to select people into your graduate program, you know. Now, coming back to planet Earth on the genes, here we have a gene significance measure which I know is a student t-test, and therefore personally, because I'm a statistician, I know that when this value is larger than 2, then this is a significant gene. T-test bigger than 2 means p-value 0.05. And when I look at the black module, notice none of the genes have a high gene significant value. They're all not significant. I immediately know this black module doesn't matter to me. But look at other modules. Example, the brown module actually contains some genes that have a gene significance of 5, p-value 10 to the minus, I don't know what, very significant. So immediately I know this is an interesting module. By the way, this one too, you know, there's several modules that have high gene significance. Yes?
I guess what I'm confused about is how are you getting the t-test? What is the comparison?
Oh, it came from having a clinical trait. I want to say body mass index. So you form a regression model, a simple linear regression, and you get a student t-test as an example. So this, it's a good question though, because every network analysis studies a different thing. Some people study body mass index, some people study autism disease, you know. And so as a data analyst, you need to define the gene significance measure that you care about, you know. It really depends. These, yes, but then you can always plot gene significance versus intramodular connectivity, and so these plots can tell you that, wow, here are several genes, you know, that are hubs, but they also have very high relationships to body mass index. And so if you cared about studying metabolic syndrome or so, these could be exciting genes, and then you would select them, you know. Other questions? Or? What?
Could be a genotype if you have like gene expression data that comes from two different genotypes.
Absolutely. Let's say you had a linkage analysis and you found this one SNP, and you just want to know, does this SNP affect certain modules? You can certainly define a gene significance measure based on the SNP, you know. Mm-hmm. These tools are very generic. It's like the student t-test, you know. The t-test can be applied to anything. Similar WGCNA. Whatever you study, but you need to couch things often into a gene significance measure, you know. Why? Because you need to have a way to decide which modules are interesting and which are not, you know. Otherwise you just have clusters, and then you say, okay, fine, here they are, you know. All right, so just to remind you, I taught you all of these steps now. I taught you how to form a network, how to identify modules, how to relate modules to external information via gene significance measures. I taught you this, how to calculate intramodular connectivity. What I didn't teach you is how to study module preservation, and we do this after the break, you know, then this will be an important topic. Let me mention one example, brain cancer, finding genes that relate to brain cancer, survival. Actually, all of these plots I showed you come from this study with Paul Michel, where we had brain cancer samples. We had two data sets. We had 55 brain tumor samples in the first set and 65 in the second set. And so we applied these algorithms and we found 5 modules. And then for each gene, we needed to define a measure of gene significance. So how did we do it? We used a Cox regression model to relate each gene with cancer survival time. And then we got a p-value, and we take minus log of this p-value per gene to define a gene significance measure. And so what we then found, so then we have a gene significance measure, and now we can form the average gene significance per module. And what we found is that this brown module contained a lot of genes that relate to cancer survival time. Clearly, it's an interesting module. The next thing is we then plotted intramodular connectivity on the x-axis versus this minus log of the p-value, and we find a relationship. And what do we know? There are some hub genes that are highly related to cancer survival time. Clearly interesting genes. And we find this relationship not just in the first data set, but also in the second data set, you know. So therefore, to find exciting genes, we did two things. We focused on genes with a significant Cox regression p-value, and also high intramodular connectivity in the brown module. And we then said, we did this in the first data set and said, well, how many of these genes remain associated with survival time in the second data set? And so let's say we started out with these genes in the first data set, 67% validated in the second data set, which may not seem impressive to you, but it is hugely impressive, unfortunately. So when, unfortunately, with genomic studies have often very poor validation success, right? You have one data set, you find very significant results. In the next data set, they don't validate. For example, if you use the 300 most significant genes in the first data set and ask how many of them validate in the second data set, only 26% of them validated, which tells you why gene expression data and genomic data have a bad name, things don't validate. However, if you focused on hubs in the Brown module, 67% validated. And so that's encouraging and may surprise you, but it's actually a very reasonable statement because this module relates to cell proliferation. And so if people who study cancer know that when the cancer continues to grow, when the genes continue to proliferate, then your prognosis is worse off. So yes, you will die. And therefore, by selecting genes that are part of this Brown mitosis module, you really stack the cards in your favor that these genes will validate, Yes? Oh, actually, we defined the module here. We did, and then we just colored the points in the second data set using the first module colors. But this is actually a very important question that you raise. How do you get modules based on, let's say, 2 or more data sets, which this is under the heading consensus modules. And so later today, I'll give you some very good R code for doing it. For example, we have an application where we had 8 different lung cancer data sets and then found modules in all 8 data sets combined, consensus modules, and they turned out to be important, All right, so another thing you could ask is, well, what is known about the hub genes in this Brown mitosis module? It turned out that 5 of the top 6 hubs were all known cancer targets. And then the 6th gene, at the time, little was known about it, but then my colleague Paul Michel validated that this 6th hub, the ASPM gene, really related to cell proliferation. So after you use siRNA-mediated inhibition, the cell lines stop proliferating. So it was a validation of that. Yeah. Another case study is afforded by looking at a comparison of human and chimpanzee brains. This was done by Mike Oldham and Dan Geschwind. So as you know, how the brains between Homo sapiens and chimpanzees, they're quite different. But you may also know that we have high genomic similarity with chimpanzees, And so it's a long-standing hypothesis that what changed is not so much genes, but there were changes in gene regulation. And of course, microarrays or gene expression studies are very good at capturing these changes. So let me show you some interesting scatter plots. Each dot here is a gene. And here the x-axis shows the mean value of a gene in human brain tissue, and the y-axis shows the mean value of the very same gene in chimpanzee brain tissue. And what do we see? We see almost a perfect, correlation 0.93, And so as a data analyst, you look at it and you say, oh my God, there's nothing different, other than noise, so the same mean expression values. However, the story changes a lot when you look at connectivity. So remember whole network connectivity. When you correlate connectivity in the human brain versus chimpanzee brain, the correlation is 0.6, much reduced, which could indicate that, the connectivity pattern changes, meaning maybe gene regulation is different. And so when you detect differences in connectivity patterns, they reflect differences in module structure. And so therefore, Mike Oldham first applied module detection in the human brain data, and he found, well, these very distinct branches and these different colors. And similarly, he had these chimpanzee brain data. And so you can then color each gene here by the human color. And you notice that many of the modules are preserved, actually. There's this large turquoise module in human brains, and it corresponds to the large turquoise module in chimpanzee brains, and so on. But later, we will study in great detail which modules are preserved from human to chimpanzees using our module preservation statistics. In any event, coming back to what's the meaning of the module. So we found this big module, the turquoise module, and you can ask, well, what's known about it? By plotting the module eigengene versus the individual samples and brain regions, we notice that, look, there's a lot of red here, meaning the turquoise eigengene takes on positive values. And so what we notice is the turquoise module correspond to genes that are overexpressed in visual cortex. Similarly, the yellow module corresponds to genes that are overexpressed in the caudate nucleus region, And so we arrive really at a nice interpretation that modules correspond to brain regions, which is expected in hindsight, of course. You have brain tissue, of course, you get large modules reflecting brain regions and structures. So conclusion, we found that gene expression values are highly preserved, but co-expression values are less preserved. And then Mike Oldham looked at species-specific hub genes, intramodular hubs, that were no longer highly so in other words, hubs in human brains that are not hubs in chimpanzee brains, and vice versa. And he found that often these differential connectivities could be explained by splicing variations and so on. So it validated it in some shape or form. So all of the material I presented today is, of course, posted on our webpage, and in particular the software. And so what I will do shortly, I will then proceed to a software tutorial where we go through some R code that so I show you how to arrive at these figures. I also mentioned there is a book on weighted networks, and if you go to the Springer webpage while you're on campus, you can download a free version of the book as a PDF file. But you have to be on campus because you need a subscription to Springer books. If you're at home, then suddenly this book will be unaffordable. Yeah. So I also want to mention many of my collaborators. So Peter Langfelder, who I referred to several times, is the first author on many of these articles. And then many students and postdocs have worked on it. I have many colleagues in neuroscience, in cancer, and mouse genetics. So Dan Geschwin, Giovanni Coppola, Roel Ophoff, Paul Michel, Stan Nelson, Jake Lucis, and so on. All right, so I would say we take another break, and then we, here, by the way, I added some citations if you love to read formulas and math and stuff. But also there are a lot of applied papers. Unfortunately, I don't have access, I guess, to, do I have access? Let me see. Okay, good. Let me just mention briefly how to find material. So what you do is you do a Google search for either weighted network or WGCNA. So I put in WGCNA or weighted network. Okay, there's a link called Welcome to the Weighted Gene Co-expression Network page. And the page is organized as follows. There's some workshop material, some tutorials and so on. But there are really two main sections. There's a theory paper section where some of you who are the bioinformaticists, you can learn about the details of the methods. And now for each article, we try to create a PowerPoint presentation. So you just click on the PowerPoint presentation. Or you could also find a web page where we post the data and all the R files, so you can reproduce many of the figures in these papers. I also mentioned as you scroll down, there comes now a section on applied papers, where we, for example, this brain cancer application is in this paper. And so you could click here, and then here would be all the microarray data, and then there are software tutorials that give you the gory details how I found the modules. There are many papers that describe mouse genetic applications. As we scroll down, we come to more recent stuff. For example, here we studied HIV. Then, as I mentioned, this is a paper on songbirds. Here are some papers on studying relationships between human brain and blood tissue, aging effects and methylation data. A lot of neuroscientists use it, the Allen Brain Atlas and so on. So I often, as I mentioned, we have the data available, so you could just reanalyze this data using your own favorite tools. And with that, I stop, and I guess I'll see you in like 5 to 10 minutes for the R software tutorial.